Hi everyone, my name's Anna Victor, and my project is on the applications of gene editing in neurodevelopmental disorders, as well as the ethical concerns of these gene editing technologies. So before we start, I want to tell you guys a little bit about me. As I said, my name's Anna Victor, and my mentor at Polygence is Rami Major. And together we wrote a project, my review paper on applications of gene editing in neurodevelopmental and degenerative disorders. A few more things about me is that I'm from the Bay Area, California, and I'm a sophomore in high school. So there's two main reasons that I decided to do this project. The first one being that I'm really interested and fascinated by genetics and neuroscience. And the second one being that I volunteer at a children's rehabilitation hospital with one of the clubs I'm part of at school. And I've seen kids who have neurodevelopmental disorders, and I wanted to see what role genetics plays in this and what possible treatment options they may have. Okay, so before we talk about these different gene editing technologies, I want to give a little bit of background because otherwise this can get really confusing. So DNA is essentially like an instruction manual for like the cell in every living organism. And the sequence of nucleotides, which are the little like stick things you see here, those determine how proteins are created. So there's two main processes to how proteins are created. The first one is transcription, where a protein called RNA polymerase binds to the DNA and essentially creates half a strand of DNA with some molecular differences called messenger RNA. This then leaves the nucleus and it will bind to another protein called a ribosome. The ribosome will read the sequence of nucleotides, which are like the blocks that you see right here, and it will string together amino acids to create an amino acid chain. This amino acid chain will then fold together and it'll create a protein. So DNA is transcribed into RNA, RNA is translated into proteins, and these proteins will go on to perform some sort of cellular function, which will then determine the phenotype or physical characteristic that a cell has. So this is kind of like a domino effect. So if there's a mutation here in the DNA, it's going to affect everything down the line. And sometimes these mutations can be harmful, which can result in genetic diseases. One example is Rett syndrome. So Rett syndrome is caused by a single gene mutation at a sequence called the MECP2 sequence on the X chromosome. So this mutation results in a deficiency of the MECP2 protein, and the MECP2 protein is essential for proper neuronal function. This is a rather rare disease as it is found in only 7.1 out of every 10,000 females. I want to put emphasis on females because this disease is predominantly found in females, and I will go into more depth as to why in a second. Individuals appear normal at birth and they hit all their major milestones, and soon after there's rapid regression. Symptoms can include loss of motor skills, speech issues, respiratory dysfunction, and seizures. And some of these seizures can be fatal and can result in early death as soon as age 20. And I'm going to be talking about different gene editing technologies specifically for Rett syndrome, but I want everyone to keep in mind that these technologies can be applied to other genetic mutation caused disorders and diseases such as Fragile X syndrome and Huntington's disease. So I talked a little bit about why that Rett syndrome is predominantly found in females, and this is essentially because females have two X chromosomes, whereas males only have one. So even if there's a mutation on one X chromosome, females would likely still have another X chromosome with the proper gene sequence. But in males, if they have this mutation, then that's the only MECP2 sequence, MECP2 protein that they're getting created. It's just the mutated protein, meaning that they die as early as like before birth or in early infancy. One more thing that I want you guys to keep in mind is that in females, there's something called X inactivation, and each cell essentially chooses one X chromosome to silence and one to keep working. And the one that's silenced will not be used to create any proteins. And this is important to keep in mind when I'm talking about DNA methylation in a second. So Rett syndrome is a pretty serious disease. So what can actually be done to help treat it? Well, I've identified three major types of gene editing that pose a means of treatment for individuals suffering from Rett syndrome. The first is CRISPR, and CRISPR essentially is like a molecular scissors. It creates two breaks in the DNA, And in the study that we discussed, they provide a template of the correct sequence, and that will be fixed. So they just get rid of what's wrong and they insert what's correct. The second method that I'm going to be discussing is DNA methylation, and I talked a little bit about exon activation. In the study discussed, the DNA methylation enzymes will go in to the silenced X chromosome that has the correct MECP2 sequence, and it will remove a methyl group, which will allow for the DNA to be transcribed. So this essentially controls the expression of the gene. The third method is RNA editing, and this basically alters the nucleotides, the sequence, after transcription, and this is safer because you're only editing what's already mutated. You're not editing like the entire DNA and having the risk of off-target effects. So this is an overview of all three studies. So the first one is DNA methylation. So they deliver this via lentiviral transduction, and this is essentially a viral delivery method. And we all know that viruses are good at getting into the cell, so they use that to their advantage. And they insert this into a stem cell, which is basically a cell that can change into any kind of cell. They're important like repair processes in the body for tissues, but they can turn into like whatever you want them to be it a blood cell or a brain cell, which in this case is what they want. So they remove the methyl group, allowing for expression of the correct MECP2 sequence. And then they observed that the neurons, which are brain cells, had like rescued phenotypes. They weren't like mutated anymore. For example, the soma size is small in Rett syndrome neurons, but after they edited it, the soma size was normal again. The second method is RNA editing. Again, they used a viral delivery method, and this method basically alters one nucleotide. So they get rid of the wrong nucleotide and they insert the correct nucleotide. And this treatment was inserted into mouse brain hippocampuses. And basically they observed that the mice had prolonged lifespan and their prolonged pauses of breathing were cured. And then the final method was CRISPR, and they delivered this through a magnetic core shell nanoparticle, which basically just helps it get into the cell faster. And this was inserted again into stem cells like the first method I discussed. And so they created the double-stranded break. As you can see, they completely separated it, and then they inserted the correct sequence. And then they observed that, again, the neurons were fixed. So going a little bit more in depth to what the effects actually were of the treatments, first we're going to talk about CRISPR. So when they used the editing method of CRISPR, they saw that there was a 36% increase in the correct production of the MECP2 sequence. They also observed that the neurons were actually like rescued from their Rett phenotypes. So it was making a difference on more than just the protein level, it was making a difference on the cellular level. For example, the soma size of the soma size of the Rett syndrome neurons are rather small, but after being treated, they're getting closer to the wild type, which is the gray. And it's the same case for the neurite length. The original is rather small, but after editing, the size is getting closer to the wild type. And then up here, you can see the dendrite complexity. So Rett syndrome neurons are not as complex in the way that the dendrites stem out, but after being repaired, the neurons look more similar to the wild type. So this basically just proves that the treatment and the production of the correct protein is actually making a difference. The second method that I'm going to discuss is DNA methylation, and essentially what they did is they wanted to see if this was resulting in off-target effects, and they saw that all the binding was occurring where they wanted it to, so little to no off-target effects. And then the phenotype changes. The wild type has a soma, but the mutated neuron does not, and when corrected, it looks like the wild type again. Finally, I'm going to talk about RNA editing, the phenotypic changes. And basically, mice with Rett syndrome only survived to 11 weeks, but once treated, they could survive more than 16 weeks. And respiratory function was also fixed because before, Rett syndrome mice had prolonged pauses of breathing, but once treated, their like frequency was more normal. So this basically just shows that what they're doing is actually making a difference in the organism. Finally, I'm just going to discuss some current issues in ethics. Some pros of the treatment are that of these all the any gene editing treatment is that they have the ability to treat diseases that may not have other treatments. Like we discussed, Rett syndrome does not have a lot of other treatments. Um, they also can be applied to many different diseases with small little tweaks. Like they just have to be told where to go and they can do whatever you want them to do. Finally, they're like a more permanent solution than some other treatments. For example, Rett syndrome has a drug that's approved, but this only alleviates symptoms. It doesn't really cure the disease, but with gene editing, you're going directly to the cause of the problem and you're fixing that. However, there's other cons to this, like it can be expensive because of how detailed these gene editing methods are. There can be chances of off-target effects. There's chances for misuse. Like I'm sure we've all heard about designer babies and, using it for purposes other than what's necessary. And there's also the issue of equity of access in different countries. Finally, I just want to talk about some things that need to be considered with each individual gene editing treatment. So one needs to consider the severity of the condition because if it's not necessary to use a gene editing treatment, then why would you do it? You also need to consider what other possible options are there because if there's other effective options, those might be better. You also need to consider the efficacy of the treatment because you don't want to insert something that's gonna harm the patient or be ineffective for no reason. And you also want to think about the benefits versus the risks, because if there's more benefit of prolonged lifespan versus dangerous off-target effects, then you want to go with that instead. Finally, I'm going to talk about some final thoughts. So I know that gene editing technology is not currently ready for clinical trials, but I think that research should be done in animals that more closely resemble humans, such as primates, so you can better assess how a human might react to these kind of gene editing technologies. I also think that editing should be given a lot of attention because it can pose a means of treatment for individuals who have diseases that have been deemed untreatable, and it gives them the chance they deserve for that treatment. Additionally, I think that perspective does need to change because in a study conducted in 2022, for only 49% of Americans were open to gene editing as a means for disease treatment. So if this is going to move forward and actually be used in humans, I think like the nation's mindset needs to be changed. Otherwise, it's never going to happen. That's all. Thank you.
Thank you so much for sharing your work. I'm so impressed by how you were able to take such a complex topic and break it down and actually make it seem clear. So I appreciate that. We don't have a whole lot of time, but I am curious. For this project, what was the most surprising discovery?
Um, I think the most surprising thing for me is that, like in the mouse models that I described, I think it was just really amazing that it made a difference in like the behavior of the mice. Like I knew it would make a difference on the protein level, but just seeing how that small little change made such a difference, like in the whole organism, and like really changed like the trajectory of the development is just really amazing.
Awesome. Well, thank you so much again for sharing everything you learned.